One of the objectives of the institute is to promote and advance the science of biology and its applications in Sri Lanka. So that IOB recently decided to conduct a series of guest talk during the month of August 2020. So far we have conducted 2 guest talks, and this is the third guest talk we are going to conduct with the sponsorship of the Bank of Ceylon, Sri Lanka. With that, I would like to introduce our guest speaker today. So it's a great pleasure and honor for me to introduce Professor S.A. Kulasooriya, one of my great teacher and the eminent scientist, who is going to talk to us about 2 days on biofertilizers, a low-cost, eco-friendly alternative to chemical fertilizers for sustainable agriculture. If I give some current and past positions of Professor Kulasooriya, currently he is working as a visiting research professor, National Institute of Fundamental Studies, Hantha, Kandy, and he is the member of UGC Standing Committee on Accreditation and Quality Assurance, and also he is the council member of the Rajarata University of Sri Lanka, Mihintale. Also, he is the member of the Board of Management, Postgraduate Institute of Science, University of Peradeniya, and he is the well-known emeritus professor of the University of Peradeniya. And he worked as a senior professor of microbiology, Faculty of Science, University of Peradeniya. Also, he contributed his service as a dean for the Faculty of Science, University of Peradeniya. And he is the Senior Research Fellow of Global Research Studies Institute, San Jose State University, California, USA. Also, it's very interesting to know that he was president of this Institute of Biology of Sri Lanka, also a founder member of the Institute of Biology. And he's the chairman, National Species Conservation Advisory Group, Biodiversity Unit, Ministry of Environment and Natural Resources. And he's the visiting professor, Department of Crop and Soil Sciences, Washington State University, Washington, USA. And if I give some, actually he has many contributions, but if I brief some contribution to the science, having returned with the PhD degree of the University of London in 1971, conducted pioneering research studies on nitrogen fixation in rice field, supported by grants from the National Science Council during a year spent at the International Rice Research Institute, or the IRRI, in the Philippines. Field studies were conducted on the application of blue-green algae, or we call cyanobacteria, and azolla for rice cultivation, and a book entitled Blue-Green Algae and Rice was published by IRRI. Continuing such studies in Sri Lanka showed the potential and limitations for the widespread application of these systems, and the report submitted won the Merit Award in the biological science in the first Merit Award scheme introduced by Natural Resource and Science Authority of Sri Lanka. In 1986, the national titular honor Vidya Nidhi was bestowed upon the author at the inauguration of the National Honours Scheme in the Sri Lanka from the mid-1980s. From the mid-1980s, the author's expertise was sought by the National Water Supply and Drainage Board to monitor algal population in reservoirs and other water bodies used for their water supply schemes, and results showed that toxin-producing cyanobacteria become dominant in freshwater resources polluted by human activities. This was published in a paper entitled Toxigenic Freshwater Cyanobacteria of Sri Lanka in the Ceylon Journal of Science in 2017. Attention was then focused upon research and development of rhizobial inoculation for food and forage legume in order to minimize the widespread use of environmentally detrimental chemical nitrogen fertilizers. These studies produce useful results. Today, local produced inoculants are provided for us widespread cultivation of soya bean, mung bean, groundnut, and vegetable beans, and the forage legume clover. By applying these rhizobial biofertilizers, it has been possible to completely replace the application of urea fertilizer in the cultivation of these crops. Sir, with this brief introduction, I cordially invite you to go with your presentation and your talk. This is over to you, sir.
Thank you, Dr. Jayalal. And I would take this opportunity to convey my gratitude and thanks for the Institute of Biology for giving me this opportunity to give this presentation entitled Biofertilizers, a low-cost, eco-friendly alternative to chemical fertilizer for sustainable agriculture. During this 45 minutes or so, I will make an attempt to show you what the National Institute of Fundamental Studies have produced over several years of research. And now we are in a position, strong position, to offer to the country alternatives that could minimize the use of chemical fertilizers. And as a matter of introduction, I will take you a little bit of your time to introduce what is soil and soil fertility. Then we move on to negative impacts of chemical use on soil and the environment, the impact of the green revolution on crop cultivation in Sri Lanka. Then we offer biofertilizers as low-cost, environmentally benign alternative to chemicals, and we will show you some pretty pictures of our results by biofertilizer applications. Soil fertility, living organisms and the effect of chemicals. Now, soil fertility is of wide scope. Generally, one can sort of introduce it as a soil that fertility that supports the healthy growth of plants. It is much more than merely providing nutrients. As you all know, soil has two major components, the non-living component and the living component. Non-living component consists of the mineral particles of the soil, stones, gravel, sand, clay, and so on. And the living component, of course, will be the macro and semi-macro and the microorganisms which are living in the soil. Now, between these two components, the non-living soil component provides the hard, solid substrate on which the plants anchor themselves and their root systems embed themselves. And of course, the other non-living components like soil water, soil air, and so on also are essential for the roots to breathe, roots to be active, as well as the roots to absorb nutrients, which are also non-living. Now, root activity depends on a healthy soil. A healthy soil should be like a sponge, not just full of clay or mineral matter. Then the living components, as we know, including microbial biodiversity, as well as other organisms, but mostly the microorganisms regulate nutrient availability and provide growth-promoting substances that are vital, essential for the growth and activity of plants. Now, continuous addition of chemical fertilizer and other agrochemicals depletes most of the living organisms, and then we end up in raising crops on dead soil. My colleague, Professor Gamini Seneviratne, has done a very simple and elegant experiment. he took some porous soil and plated it on nutrient agar full of bacterial colonies. Nobody inoculated this. This is from natural porous soil. Then he took from the same sample, he put on to the same medium, which had 100% of chemical fertilizer. There is hardly any bacterial colonies, very few bacterial colonies. Then he also added the same thing together with his biofilm biofertilizers, together 50% of chemical. Virtually, the forest soil was restored. That shows how chemical fertilizers prevent the growth of microorganisms in soil, and that is where we end up trying to raise crops on dead soil. Let's talk a little bit about the Green Revolution. The Green Revolution started a decade after the war. After the war was over in the 1950s, it came to Sri Lanka when I was a schoolboy in the 1960s. And it is the Green Revolution that made us to apply nitrogen, phosphorus, potassium, and other nutrients as inorganic mineral chemicals, synthetic chemicals in agriculture. It was introduced and popularized in Sri Lanka under the Green Revolution. Now one must realize that at that time we were adding these chemicals to soils that were full of organic matter, because we have been growing our crops for centuries using organic matter. So the added fertilizers were bound to the organic matter that was in the soil, and nutrient absorption was effective and quite efficient. Then obviously those gave dramatic results, which increased and oriented farmers toward, farmers went crazy over chemical fertilizers because they were in enormous amounts. And of course, the cultivars of the different crops were also bred for high response to fertilizer, as well as they were resistant to certain diseases. As a result, now this is about 60 years of 6 generations, 6 decades of chemical fertilizer use. Our farmers today cannot think of crop production without chemical fertilizers. We know that if chemical fertilizer subsidies are reduced or their supply is delayed, we see all over the farming areas, people come onto the streets and protest. We want fertilizer, we want fertilizer. Nobody is asking for organic or other fertilizer. They are all asking for chemical fertilizer. So they got used to it. But did these dramatic yield increases really continue? You can clearly see in this figure, we got this from the Census and Statistics as well as the Central Bank reports. There was a remarkable increase from about 35 bushels or about 2.2 or a little less than 2 tons, going up to 3 tons quite dramatically. But then it started leveling off, and today we are satisfied with the national average of 4.2 metric tons of rice per hectare. For certain areas give 6 tons, others give maybe 3 tons or 2 tons, the average works out. It does not increase beyond that. So which means that we have saturated our soils with fertilizers, but the yield increases has more or less leveled off. Let's look at what happens with the use of, or rather the misuse or overuse of chemical fertilizers. Misuse of chemical fertilizers has been a major contributory factor for the increase of non-communicable, which means non-infectious, environment-related diseases, and particularly we find them spreading rapidly among the farming communities. This is the backbone of our crop production, the farming community, and unfortunately they are the ones who are affected most by these environment-related diseases. And excessive use of phosphate and nitrogen fertilizers lead to eutrophication, excess nutrients flowing into our reservoirs, irrigation reservoirs, and other stagnant water bodies, and such eutrophication results in the formation of toxigenic algal blooms. Another research area that I have worked together with the Water Resources and Supply and Drainage Board, and we have seen how toxigenic algae have become dominant after monitoring something like 63 reservoirs in Sri Lanka. The excessive use of nitrogen fertilizers has also been recorded or suspected to cause blue baby syndrome and certain cancers, particularly in the northern provinces. Because those soils are sandy and nitrates get into the groundwater, and that causes most of these unfortunate non-infectious diseases. The other thing we have to realize is all these synthetic chemical fertilizers are imported to this country, and they are draining our foreign exchange enormously. But the government has no choice but to provide them to the farmers who are resource poor farmers, mostly under heavy subsidies. Otherwise, we will have not enough food to eat, and food security will be in danger. Therefore, we can say that both on economic as well as ecological grounds, the time has come for us to reduce our application of chemical fertilizers. Not only in Sri Lanka, there's a trend the world over, and we know last year, the Colombo Declaration on Nitrogen Management from Colombo, we declared to the entire world that time has come to reduce the use of synthetic mineral fertilizers as far as possible. Now, what I'm trying to present to you today is that we have a very attractive alternative called biofertilizers. Now, let's look at biofertilizers. What are biofertilizers? They are non-chemical. Mind you, they are live microbial entities that improve soil fertility and plant growth while continuing to live. Remember, those are the two important things. They are live and they continue to live while providing or improving soil fertility. Different from all other fertilizers, including mineral chemical fertilizers, compost, green manure, animal waste. All those fertilizers taken together are dead substrates, non-living substrates, and they provide nutrients from their own masses, so biomass, from their dead bodies. Biofertilizers are applied as live microbial inoculants, and they continue to live. And mind you, the other major difference is they are providing the nutrients from outside substrates, so external substrates. For example, we use a lot of nitrogen fixing microorganisms as inoculants, even for legumes. It is exclusively rhizobia which are nitrogen fixing in symbiosis with the legume plant. Now, they live with the plant as long as the plant lives, and they absorb nitrogen from where? It's not from their biomass that they provide nitrogen. No, they absorb nitrogen from outside, from the air. 78% of nitrogen gas is the major component in atmospheric air. And that nitrogen is converted to a utilizable form by the microorganisms and supplied to the plant. They are like a kind of a catalyst. Similarly, the biofilm biofertilizer that Professor Gamini has produced from the IFS, they grow intimately and they live together with the roots of targeted plant and enhance, accelerate, improve the absorption of nutrients from other substrates. They can be organic substrate, they can be inorganic substrate, they can be soil or anything else. They are providing from the others. So these, in fact, I'm not sure whether it's fair to call biofertilizers the term fertilizer in the strict sense. They are ameliorators, they are catalysts. Now we have already, after years of research, well over 30 years of research, produced two types of biofertilizers. Rhizobial biofertilizers for legumes, which enhance symbiotic root nodule formation, the numbers are increased, the activity is increased, and they provide nitrogen from the atmosphere. And from our field trials, we will get on to that in the course of this presentation, we have shown very clearly that we can replace the application of urea or any chemical nitrogen fertilizer 100%, completely we can replace by the application of these rhizobial biofertilizers. Then the next one that was introduced, pioneer research by Professor Gami Seniviratna, one of our colleagues at the IFS. It's a combination of 2 or more micrograms, sometimes 2, sometimes 3, or even 5, in a common matrix, common plaque or biofilm. And these form intimate association with crop roots, targeted crops, and increase the absorption of not just nitrogen, but all 3 major nutrients in nitrogen, phosphorus, potassium, and even microelement. And after doing several field experiments, it has now been shown very clearly that they can replace all these fertilizers at least by 50%, sometimes more. Now let's see how they work. Now let's get on to the rhizobium. This is a soybean plant just pulled out of the plantation. You see these root nodules, these bulbous things in the root system. Inside these root nodules live the rhizobium. And what we do is to first isolate, pick out the rhizobia from these root nodules and cultivate them in artificial culture. And after several successive isolations, this is not easy to get this stage, but after several successive cultivations, we end up with a pure culture of rhizobium in a culture plate. And then these are screened, and the best, the most active, and the most rapidly growing rhizobia are selected and then grown in crop culture in large quantities. These are 10-liter bottles, about 6 liters of broth medium. And once this cell density reaches about 10 to the power 8 cells per milliliter or more, then we inject them into modified fire dust, locally available carrier material from the fire industry, fire dust. These are sterilized by autoclaving, and the polypropylene bag, which can be sterilized, we inject the liquid broth cultivated rhizobia into those bags and allow them to grow inside the bag. It's called maturation for about 10 days. Then they are covered with another plastic cover and with the instructions. And finally, with a commercially attractive cover, we give it to provide it to the farmers. This is in Sinhala called Ranilasara Bonchi, because this is for vegetable beans. And the instructions are also given here. Now, here I should mention that different inoculants are supplied for different legume crops. Ranilasara Bonchi, or what is meant for the vegetable beans, will not work for soybean, or it will not work for mung bean, it will not work for groundnut. There are different specific rhizobial inoculants we produce for the different crops. And all what the farmer has to do is to coat this, here it is soybean, and sort of wrap them up in the coir-based rhizobium and then plant the seeds. That's all. Simple technique to the farmers. No more additions of urea or any fertilizer, and let the crop grow. And to grow and give a beautiful harvest. Here's the harvest. Here's the growth. And mind you, I'm happy to say that this extensive cultivation of soybean was done by the Government Department of Agriculture. In 2017, we provided the inoculation, and it is they who grew this and they sent us a CD of 100 photos. I have selected some of them here for this slide presentation. And this is in Dehiyatta Kandy area, we got profuse nodulation. This particular root has more than 100 nodules. It was beyond even my belief. We got profuse nodulation and ended up with a bumper harvest, I'm sure. So this was done by the Department of Agriculture, and this was the result. Then among the pioneering experiments we did in 2008, together with the cooperation and collaboration of Plenty Foods, it's a subsidiary of Ceylon Biscuits Limited, the private company, we conducted a yield response curve for soybean. This is the recommended urea fertilizer level recommended by the department is 150 kilograms per hectare. And then we use half of it, 1/4 of it, 3/4 of it, 0. And we clearly see that there's a response to application of nitrogen in terms of yield. We also, in the same experiment, we had some plots where we inoculated without any addition of urea. And we read off this curve, we found that the yield that was obtained by inoculation went even beyond the highest level of recommended fertilizer. It was nearly equivalent to about 156 kilograms. So 150 the recommended maximum. And this is 5 years later, 4 years later. So this is 2008, this is 2012. Same outsourced farmers of Plenty Foods, we got some records from them. This is the control. And here again, the fertilizer additions, also there's a response to nitrogen fertilizer, and then the response to inoculum marginally above that of the fertilizer, just like here. This was after 4 years. Now we are providing soybean inoculant for thousands of acres of farmers for the last 12 years. They keep on coming, asking for our inoculant. This is our work with vegetable beans. Here again, these are inoculated vegetable bean cultivations among the farmers' fields. And everywhere we apply our inoculant, farmers themselves realize they are growing very well. There's a nice, interesting anecdote connected with these beans. Farmers were encountering a yellowing of young bean plants after planting when they were using not our inoculant but other forms of fertilizer, including urea, poultry manure, and whatever fertilizer they had. They get some of the plants, most of the plants they claim were turning yellow and they had to replant, they die, and so on. But when we added our inoculant, we never added it for this particular purpose, but for getting rid of minimizing the chemical fertilizer. The yellowing disease has disappeared. So we don't know. Actually, we haven't done in scientific investigations. We are now, Professor Gamini and myself are writing a research proposal to get a grant, because even his biofilm biofertilizers, there's anecdotal evidence that when we use the biofertilizers, most of the diseases disappear. And according to Professor Gamini, even the brown plant hopper disease was minimized as a result of it. So there is some where we have to investigate and show, but it looks like that the addition of beneficial inoculants attract other beneficial microorganisms and the entire biome, helps the plant not only to grow and produce yield, but also to some extent make them immune to diseases. These are the empirical results from vegetable beans. In 3 locations, Hanguranketa, Killagaskada, Doragala. You see, this is the control, and there's response to nitrogen in the form of fertilizer, in the form of inoculant. And inoculant is marginally above the fertilizer recommendation. In Doragala, they are still equal. This means we can replace the addition of urea, addition of chemical fertilizer by inoculation. This is another experiment we conducted last year at Ankumbura, which is only a few kilometers away from Kandy in a farmer's field, where we did a yield response curve, just like for soybean, using vegetable bean. And this was part of a special project from a student from Ruhuna University, Kokila, I forget her surname anyway, and she also joined us. And this is my research officer, Mr. Ekanaka, helping lay out the fields. And these are the results. Here again, the yield from the inoculum was statistically not different from the maximum recommended fertilizer, which is over 200 kilograms of urea per hectare. So by adding our inoculants, we can save 200 kilograms per hectare of urea, very clearly. Then we took another record of result, that is about weed growth. Weed growth, because earlier we have observed that whenever we use inoculants, weed growth is very small, very little compared to the fertilizer additions. So this experiment, in the same experiment, we recorded the weed biomasses and calculated in terms of the 100% full fertilizer addition, we calculated, and the weed reduction where we use inoculant went up to nearly 70%, which means this is another benefit. This, of course, can be explained. This is understandable because the nitrogen that comes from the atmosphere through fixation by rhizobia is provided to the targeted crop plant and not to any other plant. Whereas when you broadcast, when you throw urea onto the soil, it is available to all the plants, crops, and weeds and everything else. Actually, the weeds will overtake the crop plants and overgrow. So this means you can minimize the application of weedicides, which is again a poison, which is polluting the environment, as well as the farmer will save a lot of labor. These are our field experiments with moonbeam, and this was done in the Tissamaharama area. Here you see the nodulation. This is other field officer, looking at the nodules with the head of the farmers. We always conduct our experiments in farmers' fields so that we are realistic, we are close to the ground, not in experimental controlled plots, but in the farmers' fields, and then the farmers themselves see the results and get convinced. Among the 16 farmers that we used to different localities and the Maharama area, you see some have got very high yield, others low yield, but nevertheless, the light blue color refers to the inoculant. Other one, the maximum fertilizer recommendation. Everywhere we got either marginally above the fertilizer or equal to the fertilizer, which means here again, we can replace the nitrogen fertilizer by our inoculants. This is our work with groundnut. Here I don't have a fully grown fields. These are the initial laying out of the groundnut fields by the farmers under our instructions. The economic is there. And then we uprooted some of the plants midway to see nodulation, even uninoculated without nectar. There are some nodulation. That is the plants are picking up the indigenous rhizobia in that soil, but it is really significantly increased by inoculation. And the results. These were done in Ampare area, in one location called Mawai Doide, and the other one in Maweli Aru in Ampare district. Inoculated pods result and the uninoculated, even here it is quite evident that we get more pods with inoculation. And these are the exact results, inoculated in blue, non-inoculated in red, and these are the number of pods you see in the and these are the seeds. In both cases, the inoculant has given a little more than the fertilizer. This is Marvel arrow. Here again, you see the difference very clearly. Means again we can replace the urea application by addition of. Now we also did some work with the forage legume clover, which is a nutritive component of animal or dairy production. And this was done at IOBSL farms. Of course, IOBSL farms can afford to put urea, no problem. They don't need cheap fertilizers. But nevertheless, this IOBSL farm is next to cotton plains. That is a prime water catchment area from where 4 major rivers of Sri Lanka start. What we want to do was to show the IOBSL authorities that you don't need to add this urea fertilizer, which they were adding every fortnight to these vast areas. But you can replace them. So we went and worked with them for 2 years. And by 2017, we were able to show that they could get better yield. Here, this is fertilizer, this is the control, similar result like earlier, response to fertilizer, response to inoculant, the inoculant, so marginally above that of the fertilizer. And this convinced the farming authorities. They stopped adding urea to those pristine water catchment lands next door to cotton plains from 2017. Now, this is how inoculation is done. Those are the experimental fields. Yeah, those are our lab and greenhouse. And this is where coated, our inoculum coated seeds are machine spread, mechanical seeding of the vast farming areas. And this is, then this, once a crop harvest is done, you can't again inoculate. What you do is then we apply liquid fertilizer, liquid crop, and then spray. And we got these results, dark green, lush growth of clover inoculated. And the root nodules, they are full of root nodules. And this is the other area, vast area where it was applied by machinery, by tractors. Again, profuse nodulation, very good. This obviously, then the farm authority didn't have to add any fertilizer, add any urea. And now that environment is safe. Another important component is to carry this across to the end users, the farmers. And that needs a tremendous amount of time, energy, and everything. Here, IOBSL is demonstrating how field inoculation should be done to the seeds and instructions given. And here we also have discussions with the local authority. This is Mr. Sivanesan, who was the regional deputy director for agriculture extension in Ampare. And interesting enough, you see, this is where we did our groundnut work with Ampare. And this is apparently former LTT militants who have been rehabilitated. They were working very well with us. And this is another interesting thing. This is bean cultivation in undulating mountainous terrain from Hangurangketa to Valley Mother. And these are bean field. Then there's a climb, another bean field, another climb. So the farmers, if they were to add urea, had to carry bags of urea on their shoulders in this difficult terrain. Whereas we give, for beans, we give 100-gram tiny packets of inoculant per kilogram of bean seeds. So they were able to carry about 20-30 kilograms in their pockets or in a polythene bag and walk up undulating terrain very easy. So that was again a clear advantage of applying the inoculants in comparison to urea application. These are the different locations where we have carried out our field trials and the different crops. This orange thing is soybean. These red ones are groundnut. And then Ambevela area, you get the clover, cowpea, and mung bean, mostly in the southern area. So we have, within our limitations, we have carried out a lot of field trials in different locations. Now we have more or less demonstrated that this is a very applicable technology. Now I come to my colleague Professor Ramini Seniratna's biofilm biofertilizers. As the introduction I said, there are two or more microorganisms drawn together in a common mucilaginous matrix. The comparable thing that most of you are familiar with is the dental plaques. Dental plaques are also biofilms, but they do an damaging thing. But these biofilms are really beneficial. Now, this is just a pictorial thing of this is a fungal filament, and around the fungal filament adhering bacteria. It's more than one species of bacteria, maybe is a fungal bacterial biofilm, what we call a combination. Interestingly enough, in addition to just growing together within this matrix, they develop special characteristics and special properties. In fact, all the activities are enhanced than when they are living on their own in isolated form. So the biofilms, in fact, form intimate links with the root system, grow with the root system, and make the roots grow better, as well as improve the activity of the roots. And this improvement of activity enables you to reduce the application of artificial synthetic fertilizer nutrients. So all three major nutrients, nitrogen, phosphorus, and potassium, can be reduced at least by 50%, sometimes more. Here are the examples. Cultivation of carrot, fertilizer treated, biofilm treated. Maize, and here clearly the biofilm with 50% chemical fertilizer gave a much better, robust harvest compared to this. This was done at the Tea Research Institute. This half the fertilizer, recommended fertilizer at 50%, full fertilizer at the nursery stage, this plant, and 50% fertilizer, and biofilm gives some equivalent to the full fertilizer. These are the biofilm results, I think, Kagal area. This is the biofilm rice hill, twice the size, and tillers compared to the fully chemical fertilizer. Potato, biofilm. Strawberry, these were part of the students' work where postgraduate students did work in Professor Gamini's laboratory. Now the science behind this, I'll try to explain. Professor Gamini would do a better job, I suppose. Anyway, this is soil fertility and the relation between microorganisms and soil fertility. Soil fertility in the center, microbes contribute, water, nutrients, fertilizers, or natural nutrients, they all contribute to soil fertility. When it comes to crop growth, microbes become the central point. And they improve soil fertility, that affects plant growth and yield, plant health, and even increase plant physiology. We have already seen that they become sometimes resistant to diseases. So the microbes here play a central role in improving plant health, soil fertility, plant growth and yield, as well as plant physiology. Now, work done with biofilm biofertilizers, initial field trials done on tea in collaboration with the TRI gave promising results, and the TRI recommended biofilm biofertilizer applications to tea nursery. Subsequent extensive field trials conducted with rice, maize, vegetables, and certain food crops, as we have seen in the illustrations, have given really benefit results with this combination, at least with 50% reduction in fertilizer. And field trials conducted last year showed that biofilm applications can increase the yield, rice yield, increase the yield from 10 to 30% with a mean value of 17%. And that is summarized here in statistical form. This is the with biofilm biofertilizer with less chemicals, and this is with the chemical fertilizers practiced by the farm. And this was in dry season 2018, 82 locations. So it's definitely representative of the different locations, 82 locations. And here are the summarized results. Yield increases over chemical fertilizer alone. We got from 7, the lowest in Ampare, to 37 in Ambalante, and the average of and all these different locations, Inoculation, Giritale, Kaudulla, Minneria, Parakama, Ampare, Niyangana, Yatta, Kandy, Ambalante, all over the island. So there's no doubt now that this biofilm biofertilizer definitely can. Right now, this technology has been, we have patented it, Professor Gamini and myself, and it has been sold to a private company, Lanka Bio-fertilizers. They are the ones who are producing and distributing and doing the marketing, everything. But right now, they are overwhelmed with the rice because the government cabinet has given the approval and recommended the use of biofilm biofertilizers for rice, and even the fertilizer subsidy will be extended to cover biofilm biofertilizers. So these are the areas where Professor Gamini and his team has done their work, representative of Sri Lanka different districts, and this 14 locations in 5 districts, you see that 90 kilograms and compared to 136 NPK can be reduced. And also about 15,000 acres in Kumragala and Ampara districts in Yala 2020. Right now, they are getting ready to extend this to another, I think, 50,000 hectares or even more. So they are, the company is overwhelmed with producing biofilm, biofertilizer for rice. At the moment, they are limited, or they are unable to provide fertilizers for the other crops, which will come in time as they get more facilities in their factories. This is the novel development by the technical assistants and technical officers of Professor Gamini. Patirana was instrumental in developing this novel technique, because earlier when biofilm liquid fertilizer was applied to the flood water of rice, didn't produce good results because the biofilm really didn't get a chance to get into the roots and adhere to the roots. So now a different technique is used where they add sand, and the biofilm, this is the biofilm, it's diluted and applied to the sand, then mixed with sand, and this is the slurry they get. And then they add this slurry together with fertilizer, urea, and potassium, I think. MOP, muriate of potash, urea, and then they make a mixture. Of course, for this already phosphorus have been added as a basal dressing to these fields. And this is after 2 weeks, they prepare and get this fertilizer together with biofilms coated, and the farmer just spread this mixture just like when he's adding fertilizer. He didn't have to change his technique. He's used to throwing fertilizer onto the fields. He's doing the same thing. And here the biofilm coated fertilizers sink and get to the bottom, to the roots, and then the biofilm takes over and goes intimately with the root system. And this is the calculated saving to the government that Professor Gamini has calculated. If biofilm biofertilizer is adopted for the entire country, the government of Sri Lanka can save 2 billion rupees worth of fertilizer subsidy annually and 6 billion in foreign exchange. Enormous. And 100 million kilograms of chemical fertilizers can be removed, which is detrimental to the environment. Chemicals that the word we use dump, so soils can be reduced. And the yield increment and the value benefit to the economy and the farmers is enormous. And considering all these, the Cabinet of Ministers, I think last month, approved promoting biofilm fertilizers for paddy cultivation island-wide. Summary of this presentation, our objective was initially to minimize the use of chemical fertilizers in crop production and reduce environmental pollution and environmental health problems. National Institute of Fundamental Studies is annually supplying rhizobium biofertilizers to about 10,000 acres of food legumes, which comprise soybean, mung bean, vegetable bean, forage legume clover, grown at Ambevela Farm. We are now working on developing fertilizers to black gram, cowpea, and Sicer, which is Padala. It also provides biofilm biofertilizers, thousands of acres. Now I think they are going for about 20,000 acres or more this season. In addition, it has been shown maize, vegetables, fruits, and other plantation crops like tea, and even rubber and coconut, biofilm biofertilizers are being field tested and giving positive results. So, if biofertilizer technologies are extended to island-wide agriculture, it could save billions of rupees and contribute significantly to the reduction of environmental health problems in Sri Lanka. And it could also minimize, therefore, enormous cost of providing health services to illnesses such as CKDU, cancers, and so on, particularly to the farming communities, which is the backbone of our food production. Thank you. These are the contact persons for our services. And of course, our team members, Rhizobium Project, Mr. Shanta Ekanayaka, Mr. Kosala Kumara, and Mr. Gamini Project, Mr. Actually should be A. Patirana, that's my mistake, Mr. Patirana. Thank you. That's the conclusion of my presentation. Thank you for listening attentively.
Okay. Thank you very much, sir. Very interesting and timely, valuable topic you have discussed with us. And then we actually gain considerably good knowledge related to that. So this after the talk, this is the time for having kind of discussion. I have received a few questions related to your presentation, sir. There's one question. It asks, what further work is required to make biofilm biofertilizer, biofilm biofertilizers mainstream?
Well, to try and answer that question, for different crops, right now, as I said, the concentration is for rice, and it is proving very well. For different crops, the formulation of biofilms changes. The composition of the components that go into the biofilm would be different. Normally, remember right, we first isolate root-associated organisms from a targeted crop. So if we want to formulate a biofilm for maize, we first isolate root-associated and rhizosphere organisms from that particular plant species. Maize, maybe the cultivated one, as well as wild-related species and so on. You have a germplasm of microorganisms, bacteria, fungi, and in the case of rice, we even want to use cyanobacteria, which are nitrogen. Then we put a combination and see which is the best formulation at the laboratory and greenhouse level. we do the screening and come out with the best formulation. I believe
Thank you, sir. Yeah, thank you, sir. And there's another question, sir. Does the efficacy of the biofilm go down with the time?
I'm not sure what it may go down with time. As long as the crop is growing, as I said, biofertilizers are live microorganisms which continue to live and function together with the targeted crop. So as long as the crop is growing, biofilm will do its job. As long as the legume plant is growing, rhizobia will do it. Once the crop gives it yield, product, and then dies, obviously the different associated organisms also will diffuse and disperse. They may not die, they may remain, but for the next round, you will have to inoculate.
Okay. So if you have, give me the permissions, I have few more questions.
Go ahead.
Okay, thank you, sir. Yeah, there's another question, sir. It asks, does the fertilizer have an impact upon the quality parameters of crops, such as tea?
Yes, I think they have already done that analysis and shown that the production improves quantitatively and also the quality and the taste. What is called these two ingredients, which give the flavor to tea. Yeah, polyphenols and the tea flavoring things have increased. And also the IFS nutritive division, we have two scientists working on nutrition of food nutrition. They have found that the nutrition of rice grains fertilized with biofilms are better, have increased, including the protein content has increased compared to fertilizers. Yeah, antioxidant is also here. Professor Gamini himself is here in the audience.
Okay, sir. Thank you, sir, joining with us. So there's another question. Has this method been used in other countries, sir?
Well, they picked up from us actually several international journals accepted that biofilm biofertilizer was introduced to the world of science by Professor Gamini's pioneering work. And now I think India has launched a project, and also they are communicating with Professor Gamini. And Lanka Biofertilizers have also taken a step to try and introduce some of these biofilms, even for large yielding tea in India, as well as I think they are also trying to introduce Bangladesh and Vietnam.
Okay.
Pakistan as well.
Okay, sir.
Four countries have started research on biofilm.
Professor Gamini just tells me that 4 countries have started research on biofilms already.
Canada.
Okay, sir.
Canada.
Brazil.
Yeah.
Then Iraq, Indonesia.
Canada, Brazil, Iraq, and Indonesia.
And India.
And India, of course, started.
Okay, sir. Okay, thank you, sir. I have one question actually need to ask. Now, considering the strains that you are using, are they endemic or whether they have some kind of specific characteristics to our country, sir?
A very valid question. We have isolated all our rhizobia from Sri Lanka. Starting point of Sri Lanka, and just a few months back, a postgraduate student from the Department of Molecular Biology and Biotechnology in the University of Peradeniya worked with us and did some molecular characterization. We have submitted a paper to the National Science Council journalists. They have undergone the first round of review, and we have replied to that. We hope to publish soon. And we have now, in molecular characterization, also identified them: bradyrhizobium, rhizobium, and cynorhizobium.
Okay, sir. So whether can they survive with somewhat harsh conditions? That means we have some different environmental conditions. So some may have adapted to dry conditions like that, sir.
Valid point. so far, everywhere this has been applied by the farmers are given good results. Results equivalent to the full addition of chemical fertilizer.
Okay.
But you can, if you can remember the slide I showed, the variation of the results among Pissamaharam with Bumbi. Some localities gave very high results, some localities gave low results. But the irony is that both fertilizer as well as the biofertilizer or the rhizobium shows the same trend. So if in a particular area, the fertilizer, chemical fertilizer also gives low yield, this also gives the equivalent low yield. So that's nothing to do with the fertilizer, but with the soil condition or some other condition, as you said, drought or any other climatic
Okay,
condition.
sir. Right, sir. Okay, sir. Thank you very much. Those are the questions we have obtained from the audience. With that, sir, I again take this opportunity to thank Professor Kulasooriya. He is one of our great teachers, as well as all the audience who joined with us. And I have to mention two more things. One is that, as I mentioned at the beginning, this is our third talk, and we have one more talk that is related to another very important area. This is on why bivalve farming is important to Sri Lanka, which also going to have with the Zoom as a Zoom meeting. This will be done by Professor WMTB Vaninayaka. He is a consultant in marine biology. So that I welcome you all to participate for this coming activity on 26th of August 2020 at 4 p.m. And I have one more announcement that now, I think many of you are familiar with the Institute of Biology and why we have this kind of organization and what are the objectives of this organization. So I welcome you all, especially undergraduate students or any other people who works or related with biology to join with us. This is the web address. Please, if you have time, go there, and there are many more things to play by yourself when you go inside the website. And with that, I conclude this very successful session. Again, thank you all the participants who have been us around more than 1 hour. Thank you very much.